Okay, so let me first thank Vijay and Vidya for inviting me here. I'm a hardcore experimental biologist, but I have a lot of thirst for theoretical biology. And I've been telling my students that they should develop a taste for theoretical biology, or at least start appreciating theoretical biology. If they don't do that, they get trapped in a local minima. They cannot come out of that local minima, and they don't get a perspective of their research work. Now, let me begin this presentation by referring to the views of Erwin Schrodinger way back in 1944. And this is what he had to say in his famous book, in What is Life? So physics is subjected to order from disorder principle, whereas an order from order principle rules biology, clearly distinguishing these two fields as separate to scientific disciplines. But today we know that biology is anything but order. Biology is full of noise, and even biologists make a lot of noise. Okay, so I'll come back to this point at the end of my talk, and this is a very important aspect that we need to study in biology. So what I would like to discuss today is how do cells of budding yeast behave in a medium containing repressing and inducing carbon sources. Essentially, I'm talking about how do cells respond when they are challenged with two opposing signals. One is glucose and another is galactose. So they signal different genetic program, and you expose the cells to these two sugars, and how does the cell respond? Not at the population level, but at the cellular level. That's the distinction, okay? Let me go back to some historical perspective. Back in 1900, Frederick Deinert made a very important observation. The observation was the following. He took yeast cells, grew in glucose, and then transferred to galactose. So what he observed was, if yeast cells are grown in glucose first and then shifted to galactose medium, there was a time lag in terms of getting adapted to galactose medium as a carbon source. That is the first observation. Second observation he made was, suppose if you grow yeast in medium containing both glucose and galactose, yeast will consume glucose first, only after that it will start consuming galactose. These are the two fundamental observations he made, and he came up with the concept of enzymic adaptation. So the credit of making this discovery of enzymic adaptation goes to Frederick Deinert. But what is strange is, after that observation, yeast was extensively used to elucidate the metabolic pathways such as glycolysis, galactose metabolic pathways, pentose phosphate pathways, and it was never used for studying the response of yeast cells for glucose and galactose, okay, for almost 100 years. However, this idea of enzymic adaptation was hijacked by bacteriologists, and they made similar observation in bacteria in the 1930s, 40s, 50s, and 60s. And this observation was made especially in E. coli. In fact, it was made in Bacillus also, but this observation was made in E. coli by none other than Monod. This is a picture taken from his thesis, where he studied a combination of sugars and a growth response. So what he does here actually, he takes E. coli, he gives both glucose and mannose as a carbon source simultaneously, and he looks at the growth, what's plotted as cell density in arbitrary units, as a function of time. As you can see here, if you use glucose and mannose, there is a monophasic growth. If you use glucose and fructose, again monophasic phase. If you use glucose and galactose, you get a small kink here. If you use glucose and xylose, you get a bigger kink. So essentially indicating that this phenomena is not restricted only to yeast, it's also observed in E. coli. So this was his initial beginning, and he went on to sort of make a major discovery using this system. So the major turning point in this whole observation was that he did not like the term enzymic adaptation, but rather he used the term enzymic induction, for the simple reason that if you use the term adaptation, it doesn't invoke any mechanism. The moment you say it's an induction, you are trying to invoke a mechanism as to how a sugar induces particular gene action. I mean, that's the idea, okay? he then went on to decipher the first secret of life. Now, this phrase you don't get in literature. I coined this particular phrase for a specific reason. After this particular discovery was made, he made another fundamental discovery of allosteric regulation. And when he made that discovery, he was so excited that he went to his colleagues and said, look, I have made a second secret of life. I have observed second secret of life. So by implication, that this observation is first secret of life. Okay? Now in my talk, I will come back to this point of genetic regulation of enzyme function as well as allosteric regulation of enzymatic function, meaning in genetic regulation, you are increasing the function of enzymes or proteins by increasing the enzymatic content or protein content. Whereas in allosteric regulation, you are modulating the enzyme function or protein function not by increasing the concentration, but by changing the conformation. Okay, so these concepts keep coming up again and again in biology, so much so, today, the number of papers published on allostery per year goes up to almost 1,500. So allosteric concept is so important. Anyway, I'll come back to this point as we go along. So, now let me just come back to yeast. What I've done here is I have listed some papers which appeared on the Diauxie of yeast. This is almost more than 110 years of the original observation of Dinard. People have started revisiting Diauxie pattern of growth in yeast in the last 5 years. There are 4 papers which appeared back to back almost 2014, 15, and 2 more papers. I'll discuss those 2 papers as I go along, one in 17 and another in 18. Simply Diauxie growth. Now, I was interested in this problem way back in early 90s, but the paradigm that I used that time was slightly different. We are talking about Melibiose, not glucose. Now, what is Melibiose? Melibiose is like sucrose. It consists of 2 sugar units linked through a bond, glucose and galactose. Okay? So yeast can also use Melibiose as a carbon source, but it has to break down this disaccharide into monosaccharides of glucose and galactose. Okay? So this was a little bit of experimentation and theory, but at that point of time, we could not continue this work. So I started doing something else. But in the recent, past few years, I again come back to this problem, and that is the point that I'm going to discuss today. Okay, now let me get back to the metabolic pathway. Don't get bogged down by the intermediates of this metabolic pathway. I have written down here. You don't have to remember this metabolic pathway, but what I want you to remember is some of the genes, GAL2, GAL1, and GAL7 and GAL10. I have mentioned it here. Now, the way the galactose gets metabolized once the cell encounters galactose in the medium is it is taken up into the cell, and you have a protein called as permease, and this is the gene designation. What it means is that yeast has a gene. We call that gene as GAL2. The number is not important. And this codes for a permease that picks up galactose. The internalized galactose then gets phosphorylated to galactose 1-phosphate galactokinase, and this is encoded by gene GAL1. Galactose 1-phosphate goes to glucose 1-phosphate by the transferase encoded by GAL7, and the UDP glucose gets converted to UDP galactose, and this glucose 1-phosphate enters glycolytic pathway. Okay? Now, in most of the experiment, we use, yeah.
These enzymes are specific to galactose, they can't
No, they're specific to galactose. Yeah. So, yeast also can use glucose as a carbon source. Yeast also can use glycerol or ethanol or any intermediates of citric acid cycle. But there's a difference between the sugars being the substrates and non-sugars being the substrates. Most of the sugars are metabolized through pyruvate and ethanol. This is what we call fermentation. You don't require a function of the mitochondria in order to metabolize the sugars. Sugars get fermented, okay? So, yeast also can utilize, in addition to galactose, it also can utilize Melibiose, but Melibiose, as I said, it's a disaccharide. It gets cleaved to galactose and glucose by alpha galactosidase, which is the gene designation for this MEL1. Now, certain species of yeast, for example, the budding yeast or the Saccharomyces cerevisiae has the ability to utilize Melibiose as well as galactose. It cannot utilize lactose simply because it doesn't have alpha beta galactosidase. There are certain species of yeast which can utilize lactose, which is again a disaccharide. For example, Saccharomyces lactis, it can utilize lactose present in the milk, and it consumes lactose in the form of galactose and glucose. So this is the metabolic pathway, okay? Now, let me come back to some few experimental details as to how we do the experiment. So what you are trying to do here is that you can take a medium containing glucose as the sole carbon source or sole energy source, and now you can monitor the expression of these enzymes involved in galactose catabolism, any one of these genes. You can either measure the transcript, or you can measure the protein, or you can measure the activity, whatever, it doesn't matter. So when you do that, you cannot detect the expression of any one of these genes, or none of these genes get expressed when yeast is growing on glucose. So glucose enters the glycolytic pathway in this fashion. And when glucose goes through this pathway, you get a lot of ATPs, and that's how the cell starts multiplying, okay? On the other hand, if you take glucose, a medium containing glucose, and then add galactose, even under those conditions, even though galactose is present, these genes are not transcribed. And that's why we call glucose as a repressing carbon source. So if you provide glucose and galactose simultaneously, first glucose will get consumed, and till the glucose concentration goes down below your threshold, the GAL genes will not be turned on. And most of the time, the GAL genes sort of get activated at the level of transcription. So to start with, if you do not have any transcripts, moment galactose is sensed, it sends a signal to genetic machinery, GAL genes are transcribed. Yeah.
Since the second part, the galactose thing is completely switched off, or there's a time delay, so it may not see as many.
No. So if you provide glucose and galactose, it consumes glucose, so much so that only after glucose comes down to a specific concentration, only then the GAL switch turns on. Yeah. But that's true for exactly. I will come back to that. Yeah. Yeah. No galactose. It's specific to galactose. Yeah. So my GAL system is here now because we have pre-grown in galactose, 100% fully active. Now if I add glucose, it comes down as a function of time. I'll show some data on this, yeah.
And then it can later become.
Yeah, again. So glucose, as long as glucose concentration, certain level of glucose concentration is maintained, GAL switch is off. Okay? So if you add galactose, GAL switch is on. Please remember that when I say switch is on, I mean the transcription of GAL genes is on. Okay? Yeah, by and large similar, right, true. This is exactly what you observe in lac system. Okay? There also it responds to glucose. Yeah, correct. Now, in addition to glucose as a carbon source, I can use a carbon source such as glycerol, which is a non-fermentative carbon source. When I say it's a non-fermentative carbon source, what I'm trying to say is that glycerol has to be metabolized only through mitochondria. It will not get converted to ethanol. It's not a fermentative carbon source, so you require oxygen for that. So glycerol enters this pathway, goes to the mitochondria, gets oxidized to carbon dioxide and water, ATP is generated and cell grows. Okay? Now, as opposed to glucose, say for example I grow the cells in glycerol, again these genes are not turned on. But to that medium, if I add galactose, the switch is on. Which means that glycerol is not a repressing carbon source, nor is it an inducing carbon source. So we call it a neutral carbon source. Okay? Which means that when the cells are growing on glycerol, the system is poised to turn the system on, ready to. The moment it sees galactose, it will turn on the GAL switch and the genes will get transcribed. Okay, so this is the system. Now, question is now, how does the cell sense galactose? How does the presence of galactose in the medium, how does it sense and how the signal is transduced all the way to the genes? I mean, that is the point that I'm going to discuss today, okay? Yeah.
Why is it glycerol, when you add galactose in the presence of glycerol, it's probably converting that to glucose and then
correct.
Doing glycolysis with it.
True. So when you, normally what is generally known is that if you provide a fermenting carbon source and non-fermenting carbon source, yeast prefers to metabolize a fermenting carbon source, not a non-fermenting carbon source. The reason for that is that for a fermenting carbon source, you don't have to have mitochondria. So when it's fermenting glucose or galactose, it doesn't have to synthesize all the machinery required for mitochondria, so it prefers a system where it can ferment and generate energy. Although, since you're asking that question, there is a slight subtle distinction between glucose and galactose. Glucose is a purely fermenting carbon source, whereas galactose is a fermenting and respiring, respiratory, so it requires some amount of mitochondria, but galactose metabolism can occur even in the absence of mitochondria. So that is one of the reasons why if you provide glycerol and galactose, it prefers galactose first, then it starts utilizing glycerol.
It prefers fermentation.
Yeah, it prefers fermenting carbon source. Yeah.
What do you mean by non-inducing?
What I mean by non-inducing is, if I grow it in glycerol, I cannot detect the expression of any of the genes. It doesn't induce the GAL system on its own, nor does it prevent galactose from inducing the system. So it's a neutral medium. I'm just trying to contrast between this medium and glucose medium, the difference between these two. So most of the experiments we do is in glycerol and we add galactose, unless you require a specific question to be addressed, glucose repression, then we start growing in glucose. Okay? Now, this is just the evolution of the galactose switch. I'm going to touch upon this quite a bit. This is the common ancestor here. After these two lineages separated, there was a whole genome duplication event. Following this, all these Saccharomyces species got evolved after this event. Okay? I'll be focusing mostly on Saccharomyces cerevisiae galactose genetic switch, but I will also touch upon the galactose genetic switch of Chloromyces lactis, which is called as a milk yeast. Its natural habitat is milk. So in the process of trying to compare these two switches, we can learn a lot about how this switch evolved over a period of millions of years. Okay? But the main focus of the talk is the GAL switch of Saccharomyces cerevisiae or budding yeast. All right. Now, let me come back to this path and let me tell you what are the elements of the switch. The question is how the signal from galactose eventually ends up at the genetic level, at the level of genes. What I've shown here is the essential elements that are required for sending the signal from galactose to genes. You have to remember these three genes. If you forget, not an issue, I've already written them on the board. Okay? So now, what are the individual functions of these three gene products? So before that, this is a regulon, which means you have a regulatory unit and all the genes are independent, unlike the cistronic or operonic structure. In operonic structure, you have one promoter that regulates the expression of the genes. But in this case, each gene has its own independent promoter. So GAL1 has an independent promoter, GAL2 has an independent promoter, right? Each gene has independent promoter, and each promoter has got two DNA cis-acting elements, what we call it as upstream activating sequences of 17 base pair each.
Does it suffice with the first?
No. Yeah. So if you have to give me an analogy, these genes are arranged in parallel, not in series. So the moment you switch on, all the lights are turned on. So if you switch off, you cannot, unless you have to manipulate the promoter, otherwise you can't do. In the natural system, you cannot do. The moment you add galactose, all these genes are turned on, despite the fact that they're all independent. Okay? No, but if you look at the proteins involved in this regulatory unit, you can make out. Okay? So this is the binding element. Now GAL4 is a protein which is a DNA binding transcriptional activator. So its job is to go and bind the DNA by virtue of the fact the promoter has these upstream activating sequences. Wherever this upstream activating sequences are there, GAL4 will go and bind. So if you grow it in glycerol, normally, GAL4 remains bound to these upstream activating sequences. So if you grow it in glycerol, although the DNA binding transcriptional activator binds to these upstream activating sequences, it cannot turn off the system because galactose is not there. So that means that you have to have a mechanism to keep the activity of GAL4, to make sure that GAL4 doesn't activate. How does it happen? That happens because of another protein called as GAL80. So if this is DNA, GAL4 is bound here, it can activate transcription, downstream gene. If I don't have any other mechanism to repress this function, it will keep activating. So that is because the GAL80 now binds. Now unless galactose is present, it cannot activate transcription. So when galactose is present, this GAL80 comes off of GAL4. Okay, how does it happen? That happens because of the presence of another set of genes called as GAL3. Now, I mentioned that the genome of Saccharomyces cerevisiae underwent whole genome duplication, and following that, a large number of duplicate genes were thrown out, a small fraction of duplicate genes were retained. One of the duplicate genes that was retained, pair of duplicate genes, was these two. Now, GAL3 is a sensor molecule which is cytoplasmic. Galactose enters the cell, and this galactose which is in the cell is sensed by GAL3, and that sensing allows GAL3 to inhibit GAL80. So when GAL3 binds GAL80, GAL80 comes off from GAL4, now GAL4 is free to activate the transcription. Probably that answers your question. Okay. That's you're talking about the lax system. There's a negative regulation, but here you have got both negative and positive regulation built into the one structure. Probably, I think as I go along, it will become clear. Why? That's why I'm trying to compare the k.lactis system and yeast system. There is some things that we can learn from that. The question why, as compared to how, is very difficult to answer in biology. All right. So this is
When you say GAL80 binds, not the protein of the cell.
Yeah. So whenever I say GAL80, I mean the protein. Okay.
And the GAL3 is present in low levels.
I'll come to that. I'll come to that. Yeah. So now what is this GAL1 doing? GAL1 is a kinase. So GAL1 and GAL3 are paralogs after gene duplication. It turned out that following the duplication, this protein lost the kinase activity. So this protein doesn't have the kinase activity now. It has only the signaling activity. Whereas this protein has this catalytic activity as well as the signaling activity. And these two activities are distinctly different. So you have one, it's a monomeric protein, you have one protein, and it has two distinct functions which are not interconnected. So I can take GAL1, I can mutate it such that it loses kinase activity, but it retains the signaling activity. So such proteins are called as moonlighting proteins. Okay? So this adds a lot of complication to the system in terms of understanding how the system functions. Okay? Chloromyces, that's why I'm going to compare the k.lactis GAL switch. Yeah, you're right. So this is the GAL switch in its most simplified form. Now I'm going to make this story a bit more complicated. Now let's talk about the basal. Yeah.
Understand the functioning. So what you say is that when galactose is present outside the cell,
Right.
it comes inside the cell,
Right.
and then binds to GAL3.
Correct.
So GAL3 is present constitutively.
Right, that's what he was trying to ask. I'll tell you that. Yeah.
Okay, so GAL3 is present all the time.
Correct.
It gets activated, it gets bound to by galactose. In that form, it then binds to GAL80,
Correct.
which was, before galactose was there outside, it was binding to GAL4 and preventing it from transcribing.
True.
But once GAL3 binds to GAL80, it makes it come off GAL4.
For the time being, let's assume that it comes off, but in the recent past, there are many controversial results. Some people argue that it doesn't come off of GAL4, rather it shifts its binding on the GAL, but that doesn't matter.
Doesn't matter.
What you're going to discuss.
Yeah, for this purpose, it doesn't matter. My question actually is that how does galactose come inside the cell?
Yeah.
What is it that's bringing it in?
I'll tell you, yeah. There are two mechanisms depending upon the concentration of galactose you use. If you use a galactose concentration below 0.05%, you require GAL2. You require this protein to be present on the membrane. But if you use a concentration of galactose above 0.05%, you don't require this. Galactose can enter by diffusion. Or it can use the glucose transporters, which are plenty. That is clearly not known whether it comes by diffusion or it uses through glucose transporters, because now you're increasing galactose concentration. Now I understand what you're trying to say. What I'm saying is, in the absence of galactose, the expression of these genes is extremely low, undetectable. But despite that, it is possible that even under undetectable levels of GAL2 concentration, there could be few molecules on the cell of GAL2, which is sufficient to take that galactose in. Once galactose in, it turns out a lot of feedback loops. Okay, is that fine?
Yeah, when glucose and galactose are present together,
Yeah.
also galactose will form.
That depends upon the context in terms of the concentration, in terms of many other things.
I have a similar question. Those galactose is not there. The concentration of galactose is separate.
No, it will not enter at all.
It will not enter.
Because unless it build, I mean, these are the questions that we'll address, because that comes under the Diauxie growth term. Okay, so this is the more complicated version of the same switch. Okay, so what we now, I talked about, so far I mentioned only the structural genes are turned on when galactose enters. Now what I'm trying to say in this slide, it is not only the structural genes, as in genes which code for enzymes, even the regulatory genes are turned on when galactose enters. So GAL3 and GAL80 are regulatory genes. They're involved in signal transduction. And even the expression of these genes is regulated by the same switch which operates or which regulates the structural genes, except for a minor difference. This promoter, GAL3 promoter, has only one GAL4 binding upstream activating sequence. This has only one. So the level to which these two genes get activated is much less as compared to the genes which have two binding sites, because of cooperativity and other phenomena. But the point I want to try to make is that the extent of expression of these genes is marginal as compared to the extent of expression of these genes. So essentially the regulatory proteins themselves are auto-regulated in response to galactose, but this does not happen for GAL4. GAL4 is not auto-regulated. GAL4 is constitutively expressed. Whereas in response to galactose, you get the expression of all the structural genes as well as GAL3 and GAL80. So this gives you a negative feedback. I mean, it's anti-intuitive. Why would a repressor concentration increase when you add galactose? So this is a positive feedback, and even GAL2 acts as a positive feedback loop. The moment a small amount of galactose enters, more GAL2 is made and more galactose is taken in. Okay? So this is the way in which the switch functions. Although I have shown that when galactose enters, GAL3 gets shifted to another conformation. This sequesters GAL80, GAL80 goes out of the nucleus. This was the old model, but we know today that even GAL3 enters nucleus, but that doesn't matter for the mechanism that we are going to discuss. Now, so this is the basic understanding of the galactose genetic switch as of today. Now I'll go back to 1948, all the way. 1948 and 1977, two fundamental observations made very early on, genetic observations, and those two observations remained as a paradox for almost 50 years. Very interesting observations. They have incredible bearing on what we are trying to understand today. Okay, yeah. So the black one is inactive form. Galactose binds, it goes to another conformation. Okay. So why does GAL-ATP regulate itself? Yeah. One of the paper appeared in 2006. What they said was that if GAL-ATP is not regulated, the cellular heterogeneity increases. So it keeps makes sure that all the cells behave in the sort of identical fashion. Now since it's a theoretical biology course, let me tell you, now that you're asking that question, we sort of modeled this whole system in 2003. Nothing was known about this system that time. This question what you're asking was not known, okay? Even the 2006 paper was not known. So we built a model on this. We asked a simple question using the model, because yesterday the discussion came up that you have a model, so what? So what do you understand by the model? So we built a model and we asked the question, what will happen if you get rid of this autoregulation? I can do it on the computer once I have a model. I remove the autoregulator loop and see what happens to the system. We asked the question, what will happen if you remove this GAL80 autoregulator loop? So it turned out, when you remove this loop alone, the system behaves in unexpected manner, in terms of output goes haywire. If I remove this autoregulator loop, the pattern of phenotype is not as expected. But if I remove both the autoregulator loops, it behaves almost like a wild type, right? But then, if that is the case, why did the autoregulator loop evolve in the first place? The answer to that is that when GAL1 got duplicated, remember the parental GAL1 had 2 upstream activating sequences. When it got duplicated, the promoter remained, the upstream activating sequence remained, so automatically GAL3 regulation still remained. In order to counteract that, GAL80 was inducted in the form of autoregulator loop. So this is our explanation. I'm not saying this is how the things evolved. But using a model, you can ask such questions and probably answer this question and get some insights into the system. That's the point I'm trying to make, okay? So why Saccharomyces cerevisiae has retained these 2 autoregulator loops? If both the autoregulator loops can be removed and you can constitutively express these 2 proteins at a specific level, that's a different question, because we have, yeah.
We talked about the
GAL1 kinase. I mean, it's difficult to answer the question, why question, but I'll tell you what happens in the system. Okay? That's the next point here.
What did you mean by GAL80
Probably let's go through some of the basic way in which the system functions. Probably that will answer your question, yeah. So this is how the system works. You take a wild type strain, add galactose, and you can monitor the expression of these genes. And this is how the increase of structural gene expression occurs, which is 10 to 100-fold. As compared to that, these 2 proteins express only 3 to 5-fold as a function of time, okay? Now, what was observed in 1948 was what will happen if this loop is destroyed. This mutant was isolated, not isolated, it was a natural variant strain was available, and they observed that this particular gene was not present, and in fact the GAL3 gene designation was given at that time. And what was observed was the following. If you take a wild type strain, it grows happily in galactose, whereas a mutant which is lacking this particular feedback loop, it takes long time to activate the GAL system. But that time, it was not known that the GAL1 functions as a redundant signal transducer. Probably that answers your question, okay? So, one of the reasons for this long-term adaptation phenotype is the GAL1 has 2 functions. In the absence of this particular signaling mechanism, GAL1 acts as a redundant signaling mechanism, and that's why you get a long-term adaptation phenotype. Now, but still this paradox still remains, because if you don't have, if you don't have this pathway, the GAL3 pathway, you cannot form GAL1, because the original signaling mechanism has gone. Under those circumstances, how this GAL1 builds over a period of time is still a paradox. Okay, so to answer your question, probably GAL1 is a redundant signal transducer. If GAL1 were not to have a signaling function, a mutation of GAL3 alone would have given non-inducibility. So that is the answer for the question. Yeah. So I'll tell you one experiment that was done in 86. You induce the system, okay? After you induce the system, you remove GAL3 function. The system remains on. It's because after induction, GAL1 has accumulated so much so that it can do both catalytic function as well as the signaling function. But so GAL3 is required for the initiation of induction. It's not required for the maintenance of the induced state. Okay, yeah. Divergent promoters. Yeah. Yeah, they're independently regulated. Yeah. Common, I mean, it's the same promoter is going in the opposite direction. Yeah. So this observation was made by Winge and Roberts in 1948. So this remained as a paradox for a long time, even after we knew that GAL1 is a redundant signal transducer. Okay. Now, I'll come to the second paradox, which was observed in 1977. This involves a little bit of genetics. So, all the strains what you are using in the system are haploid. So you have one copy of GAL3, one copy of GAL80, one copy of GAL4. Okay, this is a wild type strain. If you add galactose to this strain, it's inducible. If I have a mutation only in GAL4, this mutant was isolated in 1977. Okay, the system becomes constitutive. What do I mean by constitutive? The GAL4 gene itself is not constitutive. What I'm saying is the phenotype is constitutive. That means I don't require galactose to induce the system. If I take a strain which has a mutation at the GAL4 locus and this mutation was sequenced in late 80s, the system is constitutive. Okay, without galactose, you grow it in glycerol, system is on. The same group isolated 3 different super repressor alleles of GAL80, and these genes were sequenced again in late 1980s. I have indicated the substitutions in each gene. If a strain carries any one of these alleles, even if you add galactose, it is not induced. So it's non-inducible. Okay? Yeah.
In this GAL3 mutant, when you have this long time to turn on the GAL6, is it the whole population which is slowly turning on GAL6?
Yeah, so what he's saying is the long-term adaptation what you get in a GAL3 mutant.
Is it because the whole population is slowly turning on the GAL genes, or is it one gene which by chance had some little bit of GAL1 and then it could turn on the loops and more and more GAL1 and then it took over the population?
Yeah, I'll discuss about that in probably the second half if time permits. Okay, I'll try to address that issue. Okay, so this is the second paradox where you have this type of phenotypes. So what these authors then did, they generated haploid strains having this and any one of these alleles. No, paradox is not clear. I'll tell you what the paradox is. But before I tell you what the paradox is you make a strain which contains this combination, or this combination, or this combination. I can make these strains now. Now the paradox is, if you make this and this combination in one strain, it's constitutive, just like this. Which means that this is not able to repress this GAL4. Whereas, if you try to combine this allele with this, it becomes inducible just like the wild type, as if nothing has happened to these two. On the other hand, if you combine this with this, it is non-inducible. Now, let me clarify what the paradox is. If you say that this particular non-inducibility is because GAL3 cannot interact with this, if that is your explanation, then you cannot explain this particular phenotype. Because in order it to be inducible, this has to interact with this. So you cannot have 2 different explanations, one for this and one for this. So this remained a paradox. This is what we call allele-specific interaction for the sake of students. Allele-specific interaction in genetics immediately tells you something very interesting. This observation was made in 1977. Genes were not cloned. All the mechanisms of 80 interacting with the 4, GAL3 interacting with the GAL80 was not known. it was based on this particular observation, it was inferred that GAL80 directly, physically interacts with GAL4 based on this observation. So moment you have an allele-specific interaction, the immediate inference is that the interaction between the two genes is a direct protein-protein interaction. That is the inference. This inference was turned out to be true in late 80s, where people demonstrated biochemically that these two proteins interact. But at this point of time, it was not known. Okay, is that clear? Okay. Yeah, right. True. 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 All right. So, how wonderful that we have met with the paradox, and now we have some hope of making progress. We did not make a progress for a long time, actually. Anyway, all right. So the question now is, if you say that GAL3 doesn't interact with GAL80s-1, okay, I'll go slow, no problem, not an issue. Yeah.
The top half of the slide first.
So are you comfortable with this?
Well, this particular mutation of GAL4 seems to be one which is not repressed by GAL80.
Right, which means that GAL80 cannot bind.
GAL80 cannot bind.
Which means GAL4 is always on.
Always on, so that's fine.
Galactose.
So that line is clear. Yeah. Now, the next three lines, these are mutations, different mutations of GAL80.
Right.
And GAL4 there is the normal GAL4.
True.
And in this case, it's non-inducible, which means that probably GAL3, possibly, GAL3 is unable to affect GAL80.
Correct, but GAL80 has to bind GAL4. This mutant has to bind GAL4. If this does not happen, it will be constitutive, but it's non-inducible, which means this binds to this for sure, but this is not able to bind this.
Right.
Or this is not able to bind this.
Yes, so, and that's true for all three of them.
All three.
All three of them.
Is that fine?
So that's the conclusion from the top half of the slide.
Correct.
Now the bottom half, you look at so now you look at in turn, each of those three mutations of GAL80.
I combine with this.
And combined with that mutation of GAL4.
Right.
So the first is fine because GAL4 didn't, I mean, it's constitutive because GAL4 didn't need GAL80 to be repressed, right? So it's always active and so it's constitutive. Right. The third
is also easy to explain, where GAL3 doesn't bind GAL80s-2, but GAL80s-2 binds this.
Non-inducible.
Right. It's like this, no different.
What is
whether you have GAL4 this allele or whether you have this allele, both are non-inducible.
So non-inducible and constitutive, what's...
If you add galactose to this strain, for example, it will not turn on the system. Even if you, even if you add galactose, it will not turn on.
But you know, I would have expected that in the last line, because GAL4 doesn't need GAL80, it is also constitutive, right?
No, it's not the question of whether GAL4 needs GAL80 or not. The fact that this binds this and doesn't respond to the signal, our explanation is that GAL3 cannot bind GAL80. That's our explanation. Whereas here, if you say that this is non-inducible because this binds to GAL4, but GAL3 is not able to bind, that's why it's non-inducible. If that explanation is true, that explanation doesn't hold good here because now it becomes inducible. So that's the paradox. Yeah. So essentially this GAL80s-2 allele, it doesn't discriminate whether the GAL4 is of whether this for GAL4 allele or is a constitutive. It doesn't discriminate. That's what it means, yeah. Yeah. Can you repeat the question? Yeah. The only difference is the nature of these alleles. So this is not a wild type allele. There's a mutation where there's a... This, it's a wild type.
That one type and mutation is also in also amplified.
It is able to activate the transcription, but its behavior in terms of the ability to induce depends upon the context of these alleles.
I think if I can answer, if you look at the
So,
if you look at the fourth row, first
table, that is non-inducible. That suggests that GAL3 does not bind this particular GAL80 mutant, the fourth row, right? That's the so the highlighted one is inducible. If GAL3 cannot bind that GAL80 mutant, how can it be inducible? That is the paradox.
that's the paradox. Because you have to give two different explanations, one for this and one for this. That is not acceptable. I mean, you cannot have two different explanations to try to explain the phenomena.
We can bind to GAL80 mutant in the presence of GAL.
Right, in the presence of this specific GAL4, but not in the presence of. So, how does the difference in the allelic status of this affect this binding? That's the question. Okay? Right, true. Okay, all right. So, now what was, so this is the question. The question is how it is inducible. So, people try to do some biochemical experiments to answer this question. So, what they did was, they purified GAL3, they purified GAL80, ATS-0, ATS-1, ATS-2. All the 4 GAL80 proteins were purified. You have a wild type GAL3, you simply do a pull down assay. Those of you who are familiar with the pull down assay, it's easy to understand, but those of you who are not familiar, what you essentially do here is that you take 4 tubes, you put GAL3 in this protein, purified protein, you add 80 purified protein, you add 80 S-0, S-1, 80 S-2. Mix them and pull down this GAL3. When you pull down GAL3, whether it interacts with the GAL80 or not, it will depend on that. If GAL80 comes down, then GAL3 pulls down. So question is, now you probe for the presence of GAL80 after you pull down. Okay, that's the pull down assay. So this experiment was done. This experiment, the result of this was published in a paper in PNAS 1977, and the results are here. So you have GAL3 in all the 4 tubes. You have a tag for this to pull down the complex. You incubate either wild type GAL80 or any of these 3 mutants. And when you do the experiment and try to pull down, what you observed was only GAL80 binds to the GAL3. None of these mutant alleles bind to GAL3. So the paradox got more complicated. If it doesn't bind, then how does it induce? This was 1997. Then you cannot explain the phenotype. Now this is an in vitro experiment. It has to be compatible with the phenotype, so that you cannot explain. Now, after almost 10 years, another group went on to isolate a mutation that can interact with GAL80s-1. In fact, they isolated a mutation now which can interact with ATS-1, and that's what they have demonstrated here. So even if you are not figured out, the point I'm trying to say is, biochemical experiments demonstrate that GAL3 cannot bind GAL80s-1. If that is the real result, you cannot explain the phenotype of that double mutant. So biochemical experiments are essentially not compatible with the phenotypic data. That was the outcome of this whole exercise.
So by this time was it clear that GAL4 has no role in this binding? Why was GAL4 out of the picture?
That is a good question. Now that was the problem actually. I'll come back to that. Okay. So what we did was, these strains were isolated in 1977, We had our own lab strain. We didn't have those mutations, but we knew the mutations. This is a 2008, yeah. From 1977 to 2008. So what we did was, we recreated this strain in the lab because we knew the mutations, and we made sure that the strain behaved as they were reported. So this is the wild type strain, wild type GAL80, wild type GAL4. You don't add galactose, you hardly detect any expression of the genes. You add galactose, you get a lot of induction of the GAL genes. Now, if you have this strain, you have the fold induction is less, but you get induction and you have a slight increase in the basal expression. And this was reported in 1977. So we recreated this strain in our lab because the original strain was not available, right? Now the question is, how do we reconcile this paradox, this problem? Based on in vivo experiments, we concluded that the biochemical observations are flawed because of the wrong design of the experiments. Now, I will not tell you, we did some experiments in vivo and we concluded that there was a problem with the biochemical experiments that I just showed you in the previous paper, okay? So what we did was, we went on to redesign the biochemical experiment and see what the problem is. So what we are going to demonstrate now, in fact, GAL3 binds with GAL80s-1. So the previously reported work was not correct. Now, where is the flaw? The flaw is this. This is a normal binding pattern. Suppose you take one concentration of GAL3 and keep increasing the ATS-1P. Now I'm focusing only on ATS-1P, okay, at this point of time. As you keep increasing this protein concentration, you'll keep increasing the binding, and you pull down, and you will get this much of maximum amount of GAL80 protein pull down. If your biochemical assay is not sensitive enough to detect this much of protein, you will report it as that it's not binding. Okay? Suppose if you increase this concentration to twice, now I'll get more binding for the same amount of GAL80s. Suppose if you increase 3 times, I'll get more. So if you are doing a pull down assay in this regime, and if your sensitivity of your assay to detect this protein is very low, you will not detect it, okay? So now we repeated this experiment exactly the way what the previous investigators had done. We took one concentration of GAL3 and we incubated different GAL80 proteins, and we can demonstrate the wild type binding just like the previous workers, but we were unable to detect any of the binding of these proteins. So we are sort of somewhere here now. In the next experiment, we increase this concentration twice. Now you can start detecting the binding of GAL80s-1. We increase the concentration of GAL3 further. Now I can clearly detect the binding of GAL80s-1, whereas even at this concentration of GAL3, I am not able to detect the binding of S-0 and S-2. So GAL80s-1 has a unique property of binding to GAL3 only when the GAL3 concentration is very high.
Why was the pull down not done in vivo, and why only in vitro to do all this?
Now I'll have to get back to some of the old experiments that we did. It'll take time, but we can discuss this later, okay? So you cannot generate so much of protein in vivo, whereas in vitro you can purify and dump. But the question you're asking, let me rephrase your question. The actual question you're asking is, if excess GAL3 is required for detecting GAL80s-1, under in vivo condition, where is that you are generating that much of GAL3 to turn on the system? That's the question you're asking actually, okay? Huh? No, we don't require DNA for this, no.
But in the presence of a subject, the concentration is lower. Here you have to go to very high concentration.
Correct. We cannot achieve this concentration, but you are using this observation to explain the in vivo situation, which means that in vivo the GAL3 has to be so high, otherwise the system will not be on. But there's an explanation for that. So this was the idea. Essentially the experiment was flawed, and we now know that under in vivo condition, if you have GAL4c, ATS-1, the system becomes on because of the lower interaction between GAL80s-1 and GAL4c, as compared to the wild type 80 and 4, there is higher basal expression here. Because of these two mutations, the basal expression of GAL1 as well as GAL3 goes up. Because now we have two proteins, GAL1 and GAL3, both the basal expression goes up. That increase in higher amount of GAL3 and GAL1 is sufficient to interact, to pull out this and give you an expression. So it is sort of connected through the expression loop. Is it clear? So that is the idea. So, I mean, this whole idea came up with some other experiments which I have not discussed right now, but this is the idea. Now, I brought in the switch of k.lactis here. Now, k.lactis differs from the Saccharomyces cerevisiae in many ways. One of the difference between these two organisms is that you look at the way in which the upstream activating sequences are oriented on the DNA. If this is the DNA, in k.lactis, GAL4 binds here, GAL4 binds here, because the sequences are arranged on the opposite side. And because of that, you don't have a cooperative interaction between these two units. So because of that, there is a high basal expression and less fold induction in k.lactis. Whereas in a wild type yeast, as you can see here, the fold induction is very high, basal induction is very, very, very low, because these two complexes very tightly interact because they're adjacent to one another. The moment you change the interaction between these two, now the basal expression goes up here, whereas the fold induction comes down. So this is the way in which the system works. So by introducing just two mutations, you have sort of introduced the behavior of k.lactis without changing the promoter elements. You have not touched the promoter elements, okay? All right. So now what I'm going to show you from here on is that in what other way the mutated strain of Saccharomyces cerevisiae behaves similar to k.lactis, because these two organisms separated very early on. A lot of changes in promoter binding features and a lot of other changes have occurred. So we are now sort of introducing the features of k.lactis into Saccharomyces cerevisiae by introducing just two mutations. So this is the wild type Saccharomyces cerevisiae strain, which shows a rapid induction in addition to galactose. I discussed about long-term adaptation phenotype. If you delete GAL3, it takes long time to induce the GAL genes. Now I asked a question. Suppose if in this background, if I disrupt this, what will happen? Is it going to give me long-term adaptation phenotype like this, or is it going to give me this type of a response? Because in k.lactis, you don't have GAL3. You have only GAL1, which is high basal, okay? So we generated these strains, 4 strains, and this is, you spot those strains, tenfold dilution in a non-inducing, non-repressing carbon source. All strains are behaving normally. Now you do the same spotting in a specific medium. As you can see here, this grows on galactose. This, as we discussed before, shows long-term adaptation. If I incubate this plate for a longer period of time, you can see the growth here also. In this case, induction, but if you delete GAL3 here, there is no long-term adaptation phenotype. So just by changing these two interactions as compared to the wild type, you don't depend on an actual canonical signal transducer, simply because the GAL1 concentration has gone up now basal. That itself acts as a signal transducer. So now this mutated Saccharomyces cerevisiae strain behaves like k.lactis in these two features, basal expression, fold induction, as well as dependency on GAL3. We did some more experiments in terms of trying to figure out the behavior of these two strains. What is called as ultra sensitivity. What we do here is that you take these 3 strains, grow them in different concentrations of galactose for a defined period of time, such that we achieve the saturated amount of expression, and now monitor the expression of the gene output and plot this data, and it gives you different patterns. Now what you see here is this is for the wild type, that is this. The eta H value is 2.13, which means it is ultra sensitive. For a small amount of change in the galactose concentration, the output is very high. Whereas for the mutant, it's 0.82. It's sort of Michaelian. It's not ultra-sensitive. It's lost its ultra-sensitivity. You do the same thing for k.lactis. It is also not ultra-sensitive. So just by changing the interaction between GAL80 and GAL4, you have gotten rid of the ultra-sensitivity. It behaves more like k.lactis. We did this for single binding genes, but let's not bother about that. We did one more experiment where glucose repression, what happens to the glucose repression of these two strains? As you can see here, this is how we measure the glucose repression. You induce these strains in presence of galactose, fully induced. Now, at this point of time, you add sufficient amount of galactose and measure, or different concentration of galactose, and measure the output. As you can see here, glucose, yeah. Put different concentration of glucose to a strain which is fully induced. At different concentration of glucose, you can monitor the activity. Saccharomyces cerevisiae wild type, it's extremely sensitive. Even at this concentration, it's almost, it expresses only 10% of the GAL genes, whereas mutant is not as sensitive as the wild type, whereas k.lactis is less sensitive. You can do the same experiment in another way. Here also it demonstrates that the mutant strain is not as sensitive to glucose as the wild type, which is similar to k.lactis. All right, so when this work was going on, we came across few papers. This was a paper published in 2016, where what these people did was they isolated different species of Saccharomyces cerevisiae, and they did a classical Diauxie type of a growth. Okay? This is actually an experiment that is performed in probably BSc and MSc labs, where you grow Saccharomyces cerevisiae for a different period of time in a medium containing equal amount of glucose and galactose and monitor the growth. Okay? Monitor the growth here and measure the sugar concentration in the medium.
How does glucose turn off the gals?
That is a totally different story.
What's the question?
How does glucose
turn off the gals?
Yeah, so the question is, what is the mechanism of glucose repression of the GAL system? Now, that's a separate story, and in fact, the amount of information that's available is much more than how galactose turn on the GAL genes. There's a separate issue, but there are lots of proteins. But to cut the long story short, there's a protein called as AMP kinase, which is present even in humans. It senses glucose, and through that, it represses the transcriptional regulation of not only GAL genes, a lot of other genes which are under glucose repression. In fact, mitochondrial proteins all are under glucose repression. So that's a separate story altogether. Okay? But
GAL3, GAL80 must be involved because we see this.
True.
Sensitivity.
I did not mention this. GAL3 is glucose repressed. Even GAL4 is glucose repressed. I didn't want to make the situation more complicated. But to understand this, you don't require the exact mechanism of glucose repression. Okay, as long as you know glucose represses this. Now, this is a typical sort of a Diauxie curve, where you grow this Saccharomyces cerevisiae in these two concentration of 1% glucose and galactose. You measure the growth as well as you measure the sugar concentration in the medium. So you get a very nice Diauxie curve, two growth phases. Okay, you take another species of Saccharomyces, it doesn't show Diauxie at all. And what you can see is the glucose depletion as the cells grow. So what these people did, moment they observed this, they took all the other members of other species of Saccharomyces cerevisiae and they determined the Diauxie pattern. Turns out, you observe the Diauxie pattern Saccharomyces cerevisiae as you go up, the Diauxie pattern is completely lost. But these are all different species. Okay? So all species of Saccharomyces cerevisiae do not respond to glucose repression as Saccharomyces cerevisiae. And this is the normal laboratory strain that we keep using. Okay? This paper came up in 2016. And they attributed this difference mostly because of the changes in the promoter region of the GAL genes. 70 to 80% of this loss of glucose repression comes from the changes in the promoter that exists in this species. Now, following this paper, again one more paper came up in 2017. What these people did, instead of looking at different species of Saccharomyces cerevisiae, they looked at different strains of Saccharomyces cerevisiae picked up from different parts of the world. Okay? And they took 28 different strains. They're all Saccharomyces cerevisiae, but they differ from one another in their genetic constitution because they grow in different probable habitat. Anyway, so they took each of these strains, they took 28 different strains. Each of these strains, they monitored a parameter called as decision threshold. Now, what is this parameter? You take a strain, grow it in 0.25% galactose, and keep increasing glucose concentration, and monitor the glucose concentration that is sufficient to inhibit the GAL expression by 50%. Okay? So this is the decision parameter. You do this experiment for all the 28 strains. You get this type of pattern. These are the different strains they obtained. These are all natural isolates, not laboratory isolates. Laboratory isolates are shown here. So you can see there is so much of variation in glucose repression among different isolates obtained from different parts of the world, different habitat. So they're getting adapted to different sugar available in that particular habitat. Okay? They did one more experiment. They translated this data into data where they measured this Diauxie length in all these 28 strains, and they plotted the Diauxie lag as a function of glucose concentration. As you can see, some of the strains do not have Diauxie lag at all. What was shown in the previous slide, in the first paper, where different species have different lag, but here, even within the same species, if you pick up strains from different regions, they themselves do not have a Diauxie lag. So they went on to map. So what is the difference between these individual strains? Turned out, surprisingly, all these different strains having different Diauxie lag, it maps to GAL3 locus. The only difference between these individual strains is a polymorphism in GAL3. Just different mutations in GAL3. That itself is sufficient to confer a change in this Diauxie pattern. No, of course, they have not given the sequence of this GAL3 gene, but what I'm saying is there appears to be a mutation in GAL3 gene in all this, but what it means is that they have different mutants of GAL3. Right, so what I'm saying is, now you can look at the flexibility of this switch. I can mutate GAL3, I can get different pattern. I can mutate GAL80, I can mutate GAL4, I can get different patterns. You don't have to change the promoter region at all. But what is interesting is there was a follow-up paper on this, where they modeled this paper. They modeled this paper and they claim this sort of exercise will help us to determine the personalized medicine, essentially. Now, what is the logic here? Yeah. Right, exactly. So what I'm saying is different. So in this particular strain, you may have a mutation at a different location in GAL3 locus. In this strain, you might have another mutation in GAL3 locus. So that mutation itself is sufficient to sort of confer a phenotypic difference of this nature. Now, why I'm saying, yeah. No, that's okay, that's done, yeah.
Or uninformed, perhaps a lot of QTL mappings would have taken place. I'm curious, are there more genes thrown up? Because all the variations that you're indicating are going back to 2 or 3 genes, which I find very fascinating in some sense.
It is. But I have not told some things. Now, since you didn't bring up this issue, look, this strain is different from this. It is different. So the background, genetic background itself is different, right? So how am I saying that in all these cases, this change in the phenotypic lag is because of only GAL3 gene? Now what they did was, they did a second experiment which I did not mention. Now that you're asking this question, let me tell you. They took GAL3 gene from here, from here, from here, from all this, and put back into a lab strain which has a common genetic background. And all these lab strains bearing this GAL3 gene different show the same pattern, which clearly tells the mutation is at only GAL3 locus. Now, this is very important because this system and the sugar sensing system in humans, there's so much of parallel. For example, we have glucokinase 4, one of the glucokinases which is responsible for insulin secretion. Now people know there are mutations in glucokinase, approximately 250 mutations are known in glucokinase, and these people having this mutation in glucokinase either suffer from MODY, maturity onset of diabetes, or they can suffer from hypoglycemia. So the idea is that if you understand this phenomena and try to develop model, if an individual bears a mutation in, let's say, glucokinase, you can develop model and probably figure out to what extent his glucose metabolism is defective based on this information. So what I'm saying is this is a lot of application in terms of understanding how sugar gets metabolized.
How do these different variants are in different geographical?
Yeah, different geography, different source, like oak tree, grapes, and these sort of things.
So why is there a common mutation?
No, there's no common mutation. The mutation, same locus is same, but mutations are different. Yeah, so his question is, why do these strains have different phenotype? Because they're coming from different locations. So probably one will have to look at the biological niche in which they're growing. And that has not been related to that. Yeah. All right. So now we wanted to continue our study on this particular mutant that we have isolated. So we did a simple Diauxie curve where you take this concentration of glucose and galactose, wild type and mutant, you do a simple growth curve as a function of time. The mutant what we have isolated now and the wild type do not show any difference if you do a simple Diauxie type of growth using free glucose and free galactose.
What mutant is this? What's the phenotype?
This is the same mutant what I have been discussing, which I discussed, GAL4c, GAL80s-1. Okay? There is no change in the Diauxie pattern. Now, I can look at, remember, when you have glucose and galactose, the cells have to respond to this, and I can look at the cells, number of cells which respond to glucose and galactose, and I can determine the population of individual cells and plot this. These are GAL positives. So as it is growing on glucose, the number of GAL consuming cells are very less. As it shifts, it goes up, the GAL positives. Okay, this is fine. Now, we did an experiment. Instead of putting free glucose and galactose, we instead gave Melibiose, which is a disaccharide, and that disaccharide has to be cleaved. For that, alpha galactosidase has to come out. It's a secretory protein. It gets cleaved, it liberates glucose and liberates galactose, equimolar. And this glucose is going to inhibit the secretion of alpha galactosidase because glucose repression of GAL genes comes into picture. Whereas galactose goes in and it has to turn on GAL gene so that alpha galactosidase comes out. So that's some sort of a tight regulation. Now, the question that I'm asking now is how would the wild type we have and the mutant that we have generated behave in this scheme. So this paradigm is experimentally slightly different than the classical Diauxie paradigm, where you dump the free sugars. Okay? When we did this experiment in two different concentrations of Melibiose. As you can see here, this is the wild type, and these are the two mutants with two different concentrations, the mutants that we have isolated. As you can see, the wild type has so much of difference in terms of the response, in terms of the lag, for 0.5% Melibiose and 2.0% Melibiose. These two are wild type strains. Whereas this is the mutant strain in 0.5% Melibiose, 2% Melibiose. So there's so much of difference. So what we did next was,
The point is there's no difference essentially in the mutant.
in the mutant. Essentially, there's no difference in the mutant, whereas wild type, there's a huge difference. Okay? What we did was, next, you look at the number of cells at the same cell density, how many GAL positives are there, how many GAL negatives are there. Now please remember, under these conditions, if you have to grow on glucose, it has to hydrolyze this. That means GAL gene has to be turned on, alpha galactosidase. The moment more glucose is made, it turns it off. It's not like the classical Diauxie. The system is extremely dynamic here. So, for example, you take the cell population here and here. Look at what is the number of GAL positives in that population, because you require both the cell types. You require a lot of glucose consuming cells. If glucose is not consumed, it will repress and GAL will not be turned on. If GAL is not turned on, glucose will not be produced. So it's a very complex sort of system. As you can see here, what I've shown here is this is the wild type. At the same cell density, number of GAL positives are very less as compared to this. So here the number of GAL positives are very high. As the glucose gets used up as a function of time, then you get more and more GAL positives, both in wild type and mutant.
Those graphs are for the 0.5% case or the 2%?
This is 0.5%, this is 0.5%, what is shown is 0.5% here. So you can determine this using simple Fax analysis, or you can determine this using simple plating these cells at different time points. So there's a clear difference between this paradigm and this paradigm. Okay, although mutant and the wild type doesn't differ in the Diauxie, classical Diauxie, it does differ in terms of the way in which it consumes Melibiose. Now, essentially what it means is that mutant behaves more like k.lactis, because I told you k.lactis is a milk sugar. Its natural trait is to consume the lactose present in the milk, and it's very adapted for lactose. The moment we mutate Saccharomyces cerevisiae, it behaves more like k.lactis in terms of disaccharide utilization, okay, as compared to the wild type.
Other question here.
Yeah.
So, are you saying that this heterogeneity in the population enables some cells to grow on the basis of the metabolism being carried out in other cells?
True, true. Remember, in this, you have both glucose utilizing and galactose utilizing. They are clonal population. The genetic background is the same. Under the same condition, the clonal population exists in two physiological states, which means there's so much of latitude for the cell to turn on the system in a different way. Although all the genes are the same, it responds in a different way. That's a classic case of genetic heterogeneity. Okay, and this genetic heterogeneity can be modulated by introducing mutations.
You mean phenotypic heterogeneity?
Phenotypic heterogeneity. I'm sorry, yeah, not genetic heterogeneity, phenotypic heterogeneity. Okay? So we need a competition now. Competition between wild type, which is a control, competition between wild type and a mutant. So you take equal number of wild type, grow for some time, look at the output. What you can see here is that if you compete wild type and mutant in the galactose medium, the mutant doesn't do well at all. It suffers from the deficiency. The wild type takes over the mutant. But if you try to do the same experiment in Melibiose, the mutant takes over galactose. Or the mutant takes over the wild type in terms of Melibiose. Okay, now let me come back to the last point. There was a paper which got published in 2007 where these people studied the evolutionary significance of the GAL switch both in k.lactis and Saccharomyces cerevisiae. So what they came up with the argument is that the ancestral switch resemble this particular structure based on what is known about k.lactis. You have promoters, upstream acting sequences present on the other side of the DNA, and you don't have cooperativity, and because of that the ancestor had the bifunctional protein 1 and 3, but it's moderately inducible and bifunctional protein. After gene duplication, these two genes got duplicated and it went through some modification, and you ended up with a GAL3 which is weakly inducible in Saccharomyces cerevisiae, and you have a bifunctional protein. So this is the way the evolution would have occurred after the whole genome duplication. So the major argument they're making is that this difference occurs mainly because of the changes in the promoter structure. So what I've been saying all the time is that in order to go from here to here, you don't have to go through the same path. I can sort of create a situation where the strain, Saccharomyces strain, behaves in this fashion just by introducing an alteration in the GAL4 GAL8 interaction. I don't have to change the promoters at all. So you don't require a change in the promoter structure in order to go back and forth in terms of the way the cell behaves. Okay? Okay. Just a last piece of data which appeared in 2017. What it really shows you is that the way the phenotypic heterogeneity occurs in yeast cells, what they have done here is they have tracked the expression of GAL1 and GAL3 in single cells. So you monitor the expression of GAL1 and GAL3 as a function of time, track the same cell as a function of time. What you can see is you grow it in glycerol, pre-grown in glycerol, then shift it to galactose medium, and this is the way the GAL3 gene gets expressed, this is the way the GAL1 gets expressed. Here you pre-grow it in galactose, and in this case you pre-grow it in glucose, then shift it to galactose medium. So you can see these are all single cell tracings. You can see the variation that you get in expression as a function, as a function of time. So there is so much of phenotypic heterogeneity in the clonal population, but what we normally do in the lab, we simply take all these cells, make an extract and estimate the protein. That gives us an average value, but in reality, individual cell, its phenotype is so different. Okay.
Sorry, go back to the previous slide. So can you also draw single growth?
That is very difficult because it multiplies now. I mean, here what they have done is I think they have used microfluidic device.
I mentioned the microfluidic.
Yeah, that you could do, but I'm not sure how this is being done. I'm not sure whether people do that. But this is sort of, I'm sorry. It really tells you the phenotypic heterogeneity that you can see in the clonal population. Okay. All right. So you want me to continue with the first paradox, or shall we?
It's 10:45 the schedule.
Okay.
Whatever is a good time to
So
take a break. If there are good starting point, we can take a coffee break.
yeah, this is starting point. That's the starting point.
If there are some questions, we can take them.
Any questions?
Could just put the previous slide.
Oh, this is molecular systems biology. Author name I can't see.
Stockwell and Stockwell. 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 Used to be a postdoc with that.
All right. Yeah. So any questions?
When substrate specificity kind of come into play with the galactose and also the gal. So if I imagine that, okay, you have a disaccharides and organisms are utilizing disaccharides. They definitely have a growth advantage in not having a very tight split chain dioxide. Because if you don't have this dioxide, they definitely have a growth advantage compared to the wild type strains which are having a very split dioxide.
Okay.
So before this, when at which point of time the substrate specificity get fixed in relation to this operon or this kind of regulatory network coming out?
So are you asking that alpha galactosides will
give some other, so it might.
No, we don't have any other sugar component in the medium. We have only alpha Melibiose. Yeah.
Some other species.
Oh, I see.
Or the ancient species which may or may not be there from the alpha galactose and the GAL3 sequences now bind specific to GAL3.
No, GAL3 is a sensor. It has no catalytic activity at all.
So it's still able to bind galactose.
It is able to bind galactose.
So
and the amount of galactose that is required to turn on the system through GAL3 is a small concentration because galactose is not consumed.
Yeah.
Whereas for enzyme to utilize galactose as a source of energy, it requires a lot of galactose. You know? Does that answer your question?
There are other species of Saccharomyces which are much ancient in evolution, at which point enzyme sequences would have been able to use the same enzyme would have been able to use multiple substrates, or is it always that specific enzymes were always dedicated to one substrate, or were there multiple?
No, you're talking about enzyme evolution.
Yeah, enzyme evolution. That in relation.
I mean, there are lots of studies as to how enzymes get evolved. I mean, one of the common notion nowadays is that the actual ancestor has a broad specificity. It gets duplicated, and goes through subfunctionalization, monofunctionalization as a function of evolution, and then it gets fixed. In fact, there are very interesting studies done on glucocorticoid receptors where they resurrect the oldest protein. Given the known sequence of the protein today, using statistical techniques, they synthesize a so-called ancestral protein which doesn't exist. Okay? Once you synthesize that protein, you take it through mutations and track the whole evolutionary trajectory of that protein. That's called as protein resurrection. That technique is quite powerful because, if we can get the enzyme sequence of extant species, we cannot get the enzyme sequence of extant species. But we know we are trying to address that issue by looking at the extant species sequence, which we cannot. So people try to synthesize and they keep tracking, and there's a lot of studies, I'm sure, on enzyme evolution also using this technique. But the common notion is that the ancestral protein are sort of broad specificity, and as they went through the evolution, it gives rise to specific.
The specificity was more broader. Would a system like the GAL3 with so many mutations not having this Diauxie must have definitely had a broad approach.
Yeah, unless one does lots of such experiments, it is difficult to make some inferences. Yeah. Any other? Yeah.
So what can you infer from these points?
No. So what the minimum you can infer from this graph is that every cell is genetically identical here. So what you're doing is you take a number of cells, for example here, pre-grow it in glucose and transfer it to the galactose medium, let's say 10 to the 6 cells, and as a function of time, you monitor the GAL3 expression and GAL1 expression in each cell lineage, right? So all of them are genetically identical, yet their expression pattern is so different. The LAC system, when people came up with the LAC system, there was a concept called as genetic determinism, right? If you have these genes, this is the output. So that genetic determinism is not valid anymore. Now, why is that there is so much of difference, despite the fact that each and every cell has the same genetic blueprint? Where is that? Where does the noise arise in the system? Why do these cells behave differently?
You've labeled the third row as transiently bimodal.
Yeah.
So the bimodality is only transient, or is it also true in the steady state?
No, bimodal as in some of the cells have low expression here. If you keep continuing,
yes,
then they go back to this switch becomes on.
On this, in this picture, if you look at the last picture on the right,
Yeah, here.
many cells don't seem to go up, right? The ones which are at the bottom,
Right. I mean,
they remain there, right?
they remain there, but if you continue, probably they will go up.
So it's bimodality only in the transient behavior, not in
Initially,
the steady
right.
state behavior.
I had a question really related to that, and then a comment. So the question was, at the single cell level, do cells switch back and forth from high to low expression?
Yes.
So one has experimental evidence of that.
I can show you in the next session what I'm going to discuss. I will show you that they can switch back and forth. Of course, from this you cannot make that inference, but you can demonstrate that.
Okay. The comment had to do with what you said regarding genetic determinism and the lack of operon. That's not quite true, because the Novick and Weiner experiment already showed.
True. Now I'm talking about a textbook type of, yeah.
Maybe we should break for tea now and come back at 11:15 sharp. Sorry?
Can you elaborate on that very cryptic comment? Cryptic half comment.
Come again?
Can you
That's why cryptic. Okay. what Novick and Weiner showed, this is in the early 1950s, that with the lac operon, so which is a system for those who are not familiar, where you give a disaccharide, lactose, allow cells, this is E. coli cells, not yeast, allow cells to grow on it. And you're able to show that so long as glucose remains unconsumed, the galactose is not utilized. In any case, the point that Novick and Weiner made while studying these experiments in detail was to show that you could clone single cells from a growth culture, from one of these sorts of S-shaped growth curves. And by doing so, and by having the appropriate concentration of lactose and glucose, ratio of lactose and glucose in the medium, you could show that when you were at, let's say, a 30% level of expression, look at maybe the upper left curve here, when you were at a 30% level of expression, it didn't mean that every cell in the population was expressing at 30%, but that 30% of the cells were expressing at 100% and the remaining 70% at 0%. So you had enormous phenotypic variation, in fact, all or none variation, in spite of these cells having been generated as members of one clone.
But it's unfortunate that despite these observations, today's textbooks still continue to convey the message of genetic determinism. I mean, in a lactose system, there are many papers which came up much later, which show that phenotypic heterogeneity. So,
And those systems,
in fact, it's very interesting. Individual cells can be balanced on the threshold, and when they do that, you can see them flipping back and forth. The very elegant experiments with GFP, where the cells turn on and off, become green and dark again, and green and dark, back and forth.
any more questions?
Yes. But the interesting point is that it does, and you can pick it up.
Okay, so let's meet at 11:15, and there's a fair bit more material at that point, I think. Yeah.